Hello everyone, today we're going to be going over part C of the August 2022 Living Environment Regents. If you haven't been following along, the last three videos that I posted were part A1, part B1, part B2. We're now on part C, which is the final part. If you want to see like a full review of this test, I suggest going to the first video labeled part A1 and watching it through, and watching all the parts through. This is part C, which is the pretty much heavily based on short responses. We're going to be starting with number 56, which tells us about Fragile X syndrome. It reads, Fragile X syndrome is a genetic disorder associated with a mutation in a gene located on a particular human chromosome called the X chromosome. The disorder results in a critical protein, FMR1, not being produced. The normal FMR1 protein helps regulate the production of other proteins that play a role in the development of nerve cells. The situation is summarized below, right? So here we have a normal X chromosome, and this is not mutated, unmutated. And when it's not mutated, normal FMR1 protein is produced, and then nerve cell development is regulated as usual, right? It gives instructions on how nerve cells should form. They form that way, you're perfectly healthy. Right here, abnormal, fragile X chromosome, right? And because it has this, it's abnormal, it has this mutation, no FMR1 protein is produced, and nerve cell development is unregulated, meaning that it's not as normal, doesn't go as planned, as it should be as normal. Whenever you have an unregulated system, that's usually harmful, right? So this is the mutation, and these are the consequence or the result of the mutation, right? 56 to 57 ask us to explain how the mutation in Fragile X chromosome affects the bodies. In your answer, be sure to what? State one specific reason why the mutated gene on Fragile X chromosome is unable to produce the FMR1 protein, right? So I'm just going to answer these 2 points, right? Point number one, we have a mutated gene. Call back to when you learned about mutations, specifically in genetics. You could have a deletion, or like a delicious mutation, pretty much where the mutation, let me just quickly draw right, where the mutation completely deletes one base or deletes an entire set of bases, right? That's a deletion mutation. There's also a frame shift. Right, so let's say that instead of reading from A to T, something goes wrong, and we now start reading from C to G, and now we're missing an entire pair of bases, right? That's a frame shift mutation, and then we also have a replacement. There's probably a better term for that, but just to save some time, I'm just going to call that, where a specific base is actually replaced out of nowhere, right? So GC, so this C could be replaced with a T, and then that could ruin the entire, or this could be replaced by an A, that could ruin the entire sequence, right? So those are all the different types of mutations that can happen in your genetic material. And it's asking one specific reason why the mutated gene is unable to produce the FMR1 protein. Well, from what I just described, right, the mutation could have deleted a specific base or specific pair of bases that codes for the creation of the protein, right? Let's just say A and T, this specific AT, GCCG, right? Let's just say this entire block of code, or this gene, right, was entirely deleted by the mutation, right? So the gene that produces, or that gene that codes for the synthesis of the protein, is deleted. That could be one reason, right? So I'm going to write that down. The mutation could have deleted the gene or set of bases that code for the synthesis, which just means the creation of the protein. Right? That's my answer to part one, right? Now, number two, or bullet point number two, asks us to explain why children with Fragile X syndrome could often have learning disabilities, including speech and language problems and intellectual disabilities, right? Well, what does this mutation do, right? This mutation means that no FMR1 protein is produced, and that means that nerve cell development is unregulated, right? So here we have learning disabilities, right? We have speech and language problems and intellectual disabilities. These are all parts of your nervous system, right? So because this protein isn't produced, and because it causes unregulated nerve cell development, you're going to have irregularities within your nervous system, which include speech and language problems, right? So explain why children would often have these learning disabilities. Well, the mutation causes unregulated development of the nervous system. Right? Speech and language fall under the nervous system and will have irregularities when the nervous system is developed irregularly, right? So what I wrote was because this mutation causes an unregulated development of the nervous system, and because speech and language fall under the nervous system, they will have irregularities when the system is developed irregularly, right? All this mutation does is it causes an irregular nerve cell development, right? Nerve cells are associated with speech and language, so that's why those two sections would be irregular, right? You could also say that nerve cell development is not regulated. You could have said that the brain cells or nerve cells that regulate speech haven't developed normally. Anything like that. This was just a very wordy explanation. Moving on to number 58. It says that the chart shows the reproductive characteristics of 3 species living in an area that has recently undergone a major environmental change, right? So explain why species C might have a greater chance of avoiding extinction in the changed environment than species B. So we're looking at C versus B, right? First thing I want to see is a method of reproduction sexual, right? So we're going to have greater diversity than something than species that reproduces asexually, right? Remember that asexual reproduction means that you have the mother or the parent cell or the parent creature is genetically the child is genetically identical to the parent. With sexual, it's a combination of both parents, right? So here, knowing that, we know that species C reproduces more frequently, and we know that they produce a greater number of offspring per each reproduction, right? So what this means, and what I'm getting at here, right, because we're reproducing quicker and we're reproducing with greater numbers, species C has a greater chance of species C is going to eventually be more diverse, right, because they're having a greater amount of offspring and they're having it more frequently, right? So that means that you have a greater chance of something varying, right? Somewhere along the lines, with one of these 20 offspring produced by just 2 parents, one of them is going to have some variation that's going to help them survive, right? So because they're reproducing quicker and because they have a higher number of offspring produced with each reproduction, there's a higher chance that some variation will enter the species, that one of them will accidentally mutate a helpful, beneficial gene, and that because that gene will be introduced, it'll be passed on, and yeah, right. Whereas because species B reproduces less frequently and has a smaller number of offspring produced, they have a smaller chance of introducing deeply needed genetic variation into their population. I remember variation is needed, right? Because it says that the living area has just undergone a major environmental change. That means that the features of the species, animals living there, are also going to change, right? So what I'm getting at with 58 is that species C will or is more likely to have a greater amount of variation because it has a faster rate of reproduction, right? And a higher number of offspring, right? Some of these because they have a higher amount of variation or a higher possibility of developing variation, some of these variations could be adaptations to the changed environment. Or with increased variation, there are increased chances of developing advantageous traits to better adapt to the environment, right? Pretty much that's all there is to that. You could also say that species C has a greater chance to pass on more favorable adaptations. You could say that that's pretty much it. All this has to do is variation, right? So state one possible reason why species A could be the most successful in surviving the environmental change. Well, let's look at species A, right? Species A is asexual, which means that they produce identical offspring and they reproduce every 2 days. Right, so that's very, 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 very fast. Even though they only produce 2 offspring, that's two every 2 days. It's going to be way more than either species B or species C will ever reproduce, right? Or have the number of children they'll have in 1 year is going to be way greater than B and the amount that B and C can have, right? So I think the biggest thing we can focus on right now to solve this problem is the fact that they reproduce asexually and that they produce identical offspring, right? And why this would be good is if even one parent developed a mutation, right, that mutation would instantly be passed on to every single one of their children, right, every 2 days, right? And if that mutation was favorable, that trait can be passed on to the species way, way quicker than any sexually reproducing species can, right? So let's just say that this father or this cell has that advantageous trait in it, right? And in 2 days they produce another 2, right? And in these 2 days, these 2 cells produce another 2, right? In just 6 days, you essentially have 1, 2, 3, 4, 5, 6, 7. You have 7 new species or new not species but organisms that have now adapted to better survive in this environment, right? So what I'm trying to get at is species A or species A's short reproductive or quick reproductive rate or rate of reproduction would ensure that any would ensure that any favorable mutations or adaptations will spread to many offspring, right? So because a species a short reproduction rate or a quick reproduction rate, um, they will be able to pass on any favorable mutations or adaptations quicker quicker to their offspring than B and C will, right? So in a sense, that'll make the entire species as a whole better adapted, right? Number 60 asks us to state one negative effect of the overuse of plastics on that one negative effect that the overuse of plastic has on the environment. Let's just read this really quickly. It says that as of 2016, Americans have used approximately 100 billion plastic bags annually. An average family brought home about 1,500 plastic bags a year. Less than 1% of those bags were returned for recycling. Therefore, most bags ended up landfill, where it takes anywhere from months to hundreds of years for them to be broken down. These growing landfills are destroying natural habitats, right? That's one negative effect of the overuse of plastic bag, right? It destroys environments. Right? Boom. It destroys environments. It harms them. Right? It makes animals mistake it for food. It harms the animals living in the environment. It causes the death of animals living in the environment. It destabilizes the environment. Anything like that would account. Number 61 asks us to explain why researchers suspect that it's an enzyme that is enabling wax moth caterpillars to break down the plastic bags. We have to read further. A group of researchers in Europe discovered that wax moth caterpillars could break down the chemical bonds in polyethylene, a polymer used to produce plastic bags and other products. Though the scientists don't know what the exact chemical the caterpillar is using to break down the plastic, they predict it is an enzyme, right? So we're asking why they suspect it is an enzyme. Well, what does this chemical do? This chemical breaks down chemical bonds. So why do they suspect an enzyme? Because many enzymes are used to digest substances. Many enzymes in your body are used to actually break down many substances and break down chemical bonds, right? That's a main use of an enzyme. Think about helicase, right? The thing that unwinds your DNA, right? That's an enzyme, and what that does is it breaks the bonds in your DNA, right, in a strand of DNA. So if this is breaking chemical bonds, right, and that enzyme is breaking chemical bonds, it's a shared function, right? So what we can answer this question by saying that many enzymes break down chemical bonds, right? So we could say that researchers suspect that it's an enzyme that enables waxmoth caterpillars to break down the plastic bags because a key feature of enzymes is their ability to digest substances and break down bonds within substances, right? Or break down substances themselves. Number 62 asks us to explain why using chemically produced caterpillars to break down plastic bag would be considered an ecologically friendly solution to the problem. So, why using the chemical produced by the caterpillars is ecologically friendly, right? So, it's produced by caterpillars, which are living creatures, right? So, one thing ecologically friendly is because it doesn't have a negative impact on the environment, right? Right, think about it. If a human wants to make a chemical, they have to go into a factory and into a lab. They have to use all these even harsher chemicals. They have to heat it up. Smoke's going to be released. It needs power. Well, with the caterpillars, right, that's just them living, right? That's them being alive, right? So because they're alive, you don't need to and because you're using the chemical produced by the caterpillars directly from the caterpillars, right? You don't need to make any new factories. You don't need to hire any new people. You're literally just using an already present biological process to help the environment, right? It's also It's also produced naturally. That's another reason why it's ecologically friendly, right? And it's not likely to be harmful or toxic to the environment because it's produced naturally, right, by caterpillars, right? So 63 says, suggest a plan of action that could be carried out in your local community, which would be a step forward to solving the plastic bag problem. What they mean by the plastic bag problem is the fact that so many people are throwing out plastic bags, the fact that so many people are using plastic bags, and that they're ending up, unfortunately, in landfill, right? So the best and easiest way to solve this issue, to stop people from throwing out their plastic bags, and to stop plastic bags from ending up in landfills, is to encourage people to not use plastic bags, right? Right? Because if you start encouraging people to not use plastic bags, instead using paper bags, guess what? They're going to use paper bags. Or they're going to use cloth bags, right? They're going to use anything other than plastic. And that's going to reduce the amount of plastic bags we consume and throw out, right? So one solution to this would be to encourage or make community members use non-plastic or paper or cloth bags instead of plastic ones, right? So you could encourage or make community members to use paper or cloth bags instead of plastic ones. That's all. Right, so number 64 and 65, based on the information below. The mining of certain metal ores, such as copper and lead, can result in the contamination of soils. Wastes from the mining process can be toxic to plants and animals in the area. It has been discovered that some species of grass are able to grow in these contaminated areas. These grass plants can actually remove some of the toxic wastes from the soil and accumulate them in their tissue. Growing these resistant grass plants in contaminated soil, having them remove the toxic waste from the environment, has suggested a possible way to clean up these areas. Describe one positive and one negative outcome of mining metal ore. Right, so obviously, one negative outcome is the fact that it destroys environments, right? A contamination of soils, right? So one negative is mining contaminates soil and harms the environment. That's one thing. Or it pollutes the environment, right? That's another thing, right? And one positive outcome, right, from ore mining is that humans humans benefit, or we benefit, by gaining valuable resources, right? So think about like all the diamonds, all the iron ore you can get from mining. Those are all very, very valuable metals, and we benefit that because we are able to extract those rare metals and that valuable iron. But one negative effect is that this mining contaminates the soil and it possibly pollutes the environment, right? So number 65 asks, explain why importing grasses to clean up mining waste areas where those grasses do not normally grow could lead to unexpected environmental problems, right? So what's happening here? What's happening here is that we're importing grasses that do not normally grow in these areas, right? What does that sound like? That sounds like an invasive species, right? They don't usually grow there, so we're introducing a non-native species that could possibly become invasive, right? So that's one unexpected environmental problem. You were introducing a non-native So you're introducing it into a non-native species into an environment where it can become invasive. right? Or you could have said that once you introduce it, the grasses would compete with native plants, which is kind of the same thing as an invasive species, right? Animals in the new area could be harmed by this new grass, right? Um, like fishes or any animal that eats grass could eat it, and because it's not their normal diet, they can get sick and die, right? So all of these, pretty much all of these are different ways to go about this problem, but I think that the introduction of a non-native species really mainly opens up the possibility of it becoming invasive and an introduction of an invasive species into an environment, right? Number 66 says that today many diseases have been linked to mutations that cause mitochondria to fail. Patients who suffer from a mitochondrial disease may suffer from fatigue and weakness. Explain why patients with a mitochondrial disease would tend to experience these symptoms, right? So what does the mitochondria do? The mitochondria, we've all heard this one before, is the powerhouse of the cell, right? What does that mean? That means that mitochondria produces ATP. ATP, right? ATP is the same thing as energy. It's the thing that we get energy from. How am I able to talk right now? It's because all my mitochondria just produced ATP that powers my mouth muscles to move, right? And it's also powering my brain to think of the next word that I'm going to say. So it produces ATP, right? Mitochondrial diseases, they suffer from fatigue and weakness, right? So if I had low ATP, if I had low energy, I would probably be weak and I'd probably be very tired, right? So, why patients with mitochondrial diseases would tend to experience these symptoms? The mitochondria produces ATP, right? Patients with mitochondrial diseases may produce a significantly less amount, or produce significantly less ATP because they produce less energy, less ATP, they have less energy to power muscles. To power their muscles and body. This causes fatigue and weakness, right? Boom. Right? Because the mitochondria produces ATP, patients with mitochondrial diseases may produce less ATP, and because they produce less ATP, they have less energy to power their muscles and body, and because they have less energy to power this, they're going to be a lot more tired, weak, and fatigued. That's all. Moving on to number 67, it asks us to explain how snow coverage affects the population of the snowshoe hare. Well, let's read it, right? Snowshoe hares are found in northern evergreen forests of the United States. The physical characteristics of the hare enable them to hunt food and hide from their predators during cold, snow-covered winters. It seems like a very important detail, right? They have large snowshoe-shaped feet and thick fur. A change in fur color during an annual molt, also called shedding, occurs before the winter season, causing white fur to replace brown fur of summer. The amount of snow cover in these northern forests has decreased in recent years. Research has shown that the decrease has a significant effect on snowshoe hare population. Even though the carrying capacity of the forest has not changed, researchers have estimated that for every 7 days that snow covers the ground, the snowshoe hare populations are 4 times more likely to survive, right? So I'm just going to go stop reading there. I'm going to go back to the questions, right? So explain how how snow cover affects the populations of the snowshoe hares, right? So didn't we just read that researchers have estimated that for every 7 days snow covers the ground, snowshoe hare populations are 4 times more likely to survive, right? The more snow cover, the higher chance the snowshoe hare will survive, right? Or the more snow coverage there is, the higher the population of snowshoe hares. You could have said that the snowshoe, the snow greatly benefits the survival of the population, right? And number 68 asks us to identify the environmental factor that stimulates the fur color of the hair to change from brown to white, right? So it's an environmental factor. So let's go back into the reading. It says that since the molt from brown fur to white fur is a response of decreasing hours of daylight in the fall and not the arrival of snow, the later the snow arrives, the greater the chance that white hares will be caught by their predators. Snowshoe hares play a major role in the stability of forest ecosystems. Their loss would affect species such as lynx and great horned owls. If the amount of snow cover continues to decrease, researchers are concerned that it'll be harder for snowshoe hares to survive in their current habitats, right? So, and we're asking for an environmental factor, right? Well, didn't we just read that that since the molt from brown to white fur is a response to decreasing hours of daylight, doesn't that mean that daylight, or the length of daylight, the length and frequency of daylight stimulates or causes, that's a synonym of stimulates, the fur color of the hair to change, right? It literally just stated here that the molt is a response to decreasing hours of daylight, right? So the length and frequency of daylight stimulates the fur and change, right? Number 69 asks us to identify a specific environmental issue that most likely affects snowshoe hare populations in northern ecosystems. Well, let's think about it, right? If the length of day matters, right? If the amount of snow matters, and so pretty much it's the length of day, right? And it's the amount of snow that really matters. Why is it the length of day? Because the longer the day, the more chance there is for the snow to melt, right? And the amount of snow, as we stated there, if there's 7 days of snow, they're 4 times more likely to survive, right? So a specific environmental issue could be global warming, right? What is global warming? Global warming is the phenomenon where the average temperature of the world is increasing rapidly, right? And because the temperature of the Earth is increasing, that means that the snow melts faster, and that means that there's less coverage of snow. There's less snow covering the environment of the hare, which greatly decreases their they're likely to survive. Their ability to survive, because they need that snow cover to survive. So you could say global warming, temperature increase, and that causes snow to melt, right? Because they melt, because snow melts, hares are more prone to predators, right? And or you could say which causes them to move further north into colder areas with more snow coverage. That's all. Number 70 says a graph below, based on 1985 data, represents the time of year when the fur color of the snowshoe hare in the northern Rockies does not match the color of their surroundings. The bold line on each graph indicates the period of time that snow covers the ground. The shaded area in the graph represents the mismatch of color. Okay, so what's happening here, right? Well, this bolded line is, what did we just say it was? So it's the period of time that it covers the ground, right? So here in 1985, there's a pretty big period of time that it covers the ground, right? And there's a very short sliver, very short sliver of time where they molt, right? In this short period, they're changing from brown fur to white fur, and now they're blended in. In 2045, however, the amount of the time frame of the snow has greatly decreased, right? So they're out of color for much more longer. They're now covered for much longer, simply because there's a smaller amount of snow coverage on the ground, right? And then finally, right here in 2075, they're really screwed, right? Because once again, the snow coverage has decreased significantly, so that for almost half of the winter, right, they're mismatched in the color, and then their fur is now visible for predators to swoop in and attack them, right? So it's asking us to identify one change in the characteristic of snowshoe hares in this ecosystem that would be most likely selected for if the trends in B and C prove true, right? So think about it. If B and C are true, which meaning that if the probability, if more and more snow melts, and there's a greater and greater mismatch of color found in the population, right? What's one trait that can evolve from this? Well, the way I'm looking at it is the more that the snow melts, the less that they really need that that ability or that the needing or like the need to quickly molt, right? So because why should they molt very quickly, right? Or why should they molt this early, right? When the snow coverage will happen like months later, right? So one thing that could change is the amount of time, the molting time, aka the specific day or the specific month that they biologically begin molting automatically, right? Another thing is that the fur color of the hair might be darker for more of the year, right? Because as more of them mismatch the color, this means that more of them are brown, right? And that means that trait is now being passed on more frequently in the environment, right? So one change in the characteristic of snowshoe hares could be their molting time. Or the period that they molt for, right? Maybe pushed forward, right? Maybe pushed forward as the amount of snow coverage is also pushed forward, right? So moving on to number 71 and 72. So this is a swollen red abdomen, apparently. It's black and white, so I can't see that. Right, so scientists have discovered a parasitic roundworm that makes its ant host look like a juicy, ripe berry. Worker ants collect materials from soil to feed larval ants. Often the soil contains roundworm eggs that are consumed by the ant larvae. The roundworms develop from the eggs and eat the ant larvae, mate, and produce hundreds of roundworms. As the roundworms develop, they cause an increased reddening of the developing ant's abdomen and take nutrients from the ants. Just as a fruit reaches peak color when its seeds are ready for dispersal, an infected ant's abdomen reaches peak redness and as the ringworm matures. Birds don't normally eat the foul-tasting ants, but are thought to eat the ants infected with roundworms since they look like red berries. The roundworm eggs move to the bird's digestive system unaffected and pass the soil in the bird's feces. So what happens? So, so a bird poops them out, right? Poops these spores out, and they're in the soil, right? The poop pretty much decomposed, but the roundworm is now in the soil, right? So it begins developing in the soil, and eventually a worker ant comes in and it collects some of the soil as a material. Some of that soil has that roundworm, right? So then the ant goes back to its home and here's all the larvae, right? I guess it spits out the dirt right next to the larvae, whatever. These roundworms then enter the larvae, right? They crawl around, they enter the larva. And now that they're inside of the larva, they pretty much eat away at the larva, right? And now some of these larvae are going to grow up to become ants, right? And some of the ringworm actually latches onto the ant and infects it. When it's inside of the ant, what happens is it pretty much steals nutrients and increases the redness of their abdomen, right? And increases the size. So if this was a regular ant, this would be one infected by roundworm, right? Until it looks like a juicy I know I'm using the opposite color for this, but until it looks like a berry, right? Up to which a bird eats it, the larva is now transferred to its poop, and it could do and repeat the process, right? So we're asked to state one reason why the roundworm is considered a parasite of this tropical ant. Well, what's a parasitic relationship? A parasitic relationship is where one benefits and the other is harmed, right? So let's just describe this, right? So the roundworm takes away nutrients from the ant as it develops. That's a parasitic relationship, right? Here we say that developing ants abdomen and take nutrients from that. They take nutrients from the ant, right? The ant does not benefit from this, right? But the roundworm does. So that's a parasitic relationship because one is being harmed by taking nutrients away, the other is benefiting. You could also said that it's a parasitic relationship because it increases the likelihood that the ant will be eaten. Once again, that's harming the other. You could say that the roundworm benefits by living in the ant, and the ant is harmed. You could have said that. We just went into more specifics. Moving on to 72, describe one advantage the roundworm has by having birds involved in parts of its life cycle, right? So why, right? Why is this necessary? Well, it's because that the birds can transport or spread roundworm eggs to new areas that could possibly have new ant populations, right? Because think about what happens, right? Once the roundworms have infected the colony, they pretty much eat all of the larvae, and eventually there's not going to be any larvae left for them to eat, and they're going to die out, right? So before they die out, they want to get eaten by the bird, so that the bird, which if you didn't know this, if you flies around. Wow, the bird flies around. When it flies around, it could fly pretty great distances. Birds can fly, a couple miles when foraging for food, right? So, and obviously it's going to poop in some time there, right? So, by getting eaten by the bird, what the roundworm benefits by the possibility of the bird flying a couple miles, pooping it out, right? And now it's in a brand new area where then it can hopefully find a new ant colony to infect because the one that it already infected is already dead, right? So one advantage it gets is that it could spread to new areas and thus survive. Part D, we're now moving on to part D, which is 73 to 74. This video is going to be really long, so I apologize. It asks us to base our answers to the questions 73 and 74 on the information below and your knowledge of biology. The photograph shows two birds on a bird feeder. Right? Studies have shown that the length of bird beaks within Sonnenberg populations may be influenced by the presence of bird feeders. When bird feeders are widely used in one area, birds were observed to have longer beaks. In an area where bird feeders were not used, the beaks of these species were of average length. One possible reason for the likely increase of the beak length is that the birds with longer beaks do what? Or why? Well, think about it, right? You should be thinking about Darwin's finches. Finches right now, the Galapagos Islands. What happened there was that the different birds evolved and adapted different beaks so that they could extract food found exclusively on their specific islands more effectively. What's happening here is that whenever there's a bird feeder, you see this little hole here. That's where the food comes out of, right? And if you, as a bird, you need energy, which you get in the form of food, to survive and then to reproduce and pass on those genes. So whoever gets the most amount of food, whoever gets the most amount of energy, has the highest chance of surviving and has the highest chance of growing and actually reproducing, right? And if you want the most energy, you want the most food. How do you get the most food from this hole? Well, you develop a larger beak, right? The longer your beak is, the deeper you can reach into that and the more food you can get, right? And because the parents with the longer beaks are going to be the ones who are surviving greater, they're going to pass on those beaks to their offspring because the beak is advantageous, right? So which choice just summarizes what I just said? It's choice 2, right? One possible reason for the increase in beak length is that the birds with longer beaks had a more successful adaptation for surviving in the area. Why? Because they could get more food, right? Right, so they needed to reach the seed with a lot of people choose 3, commonly common mistake. This is wrong because the birds don't grow longer beaks. They evolve over time, or they find an adaptation over time to grow longer beaks, right? This is Lamarckian evolution. That's wrong, right? That's like saying that that a giraffe got its long neck in its lifetime, right? Because it just wanted to reach trees. No, the giraffe got its long neck over generations of evolution because the longer neck was a successful adaptation for survival in its habitat, right? Number 74 says that the presence of bird feeders in the area would represent what? Well, it's a selecting agent, right? We just said that areas with a lot of bird feeders end up changing the composition of the environment to contain birds with longer beaks, right? So what the bird feeder is doing is it's inadvertently selecting for longer beaks. We call that a selecting agent, right? It's making sure that birds with whether intentional or not, it's making sure that birds with longer beaks get more food and in turn survive, right? So in a sense, it is selecting for longer beaks. This is called the Elephant Shrew. The Elephant Shrew spends its days searching the leaf litter on the forest floor for insect prey. When first discovered, due to structural similarities, the elephant shrew was classified with other shrews and their relatives. However, scientists recently reclassified the elephant shrew as shown in the evolutionary tree below. Right? So we have this evolutionary tree, right? We have all other mammals and we have rodents, and we have pretty much shrews and relatives were grouped with all other mammals, right? But now they've been regrouped. Rather than being on this side of the tree, they've been regrouped to this side of the tree, right? So that's a pretty big change, right? This was saying that shrews were pretty closely related to all other mammals. This is saying that they're pretty far away from all other mammals. So let's see what 75 asks us. It asks us, the new, more accepted classification of the elephant shrew is probably based on an analysis of what? Right? So the best way that you can compile one of these evolution trees is through comparison, right? Whether that be the comparison of fossils or the comparison of DNA. That's usually how these trees are formed, right? They analyze fossils of previous species, they identify similarities within them, and eventually they that's how they draw conclusions of relations, right? So we're obviously we're going to be narrowed down to choose from number 3 and 4, where a number of newly formed shrew fossils and the DNA presence in the cells of elephant shrews. Now, fossils are mainly used for already extinct creatures, right? Fossils could be used to determine how related this species was with this species, or why this split occurred and this split occurred, right? The shrew isn't an extinct species, right? It's alive, and always DNA analysis is always going to be the most accurate way that you can actually classify and identify similarities within a species. So the answer to 75 is going to be 4. The DNA present in the cells of elephant shrews, right? They took this DNA and then they examined it, and then they compared it with all other mammals. They compared it with primates, rodents, and rabbits, right? And they found that they weren't really that related. Rather, they were related more with aardvarks and golden moles, you know? So remember that DNA is often the most effective and the most accurate way of classifying something, and that fossils are usually used with extinct species, and that either way DNA would be much more accurate, right? So number 76 asks that according to the new evolutionary tree, elephant shrews are most closely related with what? Well, let's see. So whatever pretty much what we're looking at is whoever's lines touch the soonest is what it's going to be most closely related to. That's the rule that I'm going to follow here. So number 1 says manatees and hydraxes, right? So how many nodes away? How many nodes? The less nodes away you are, and what I mean nodes is this kind of this branching point. The less nodes you are away from something or someone, the more related you are to them, right? So here, manatees and hydraxes, that's 1 node. 2 nodes, 3 nodes, right? So that's 3 nodes away. Shrews and their relatives. Shrews and their relatives, well, that's 1 node, 2 nodes, 3 nodes, 4 nodes, 5 nodes. That's 5 nodes away, right? So we could automatically eliminate that because this is 3 and that's 5, right? Number 3 says Tenrecs, golden moles, and aardvarks. Let's see how many nodes away that is, right? So that's 1 and 2. That's only 2 nodes away. So 2, so far 3 is our answer, right? We can cross out number 1. And number 4 says primates, rodents, rabbits, and relatives. That's 1, 2, 3, 4. That's 4 nodes. This has the shortest distance between them. So yeah, that means that they're closest related. Number 77 asks us that, or tells us that the elephant shrew is at risk for extinction because its habitat is very limited. The elephant shrew can only be found in 2 forest locations within the country of Tanzania. Even though these locations are protected, they could be harmed by fires and human activities. Explain why it is important to continue to protect the habitat which the elephant shrew is found in, right? Well, they're at risk of extinction, right? And whenever an species goes extinct, there's a great impact that it has on the environment, right? It greatly destabilizes the environment, right? So that is one important reason to protect the habitat which the elephant shrew is found in. Because if the habitat is destructed, right, the elephant shrews will go extinct. That is a perfectly good answer here, right? It's important because without protection, the shrews may become extinct, right? You could also say that they could destroy the destruction of habitat could disrupt the entire food web. That's another reason to support that, right? So 78 and 79 are based on the information and knowledge of biology. A student hypothesized that the pulse rates of his classmates would increase after walking. The student then obtained pulse rates from 5 classmates as they walked for 15 minutes, right? So the data in beats per minute were recorded as 78, 68, 84, 88, and 90. Identify the dependent variable investigated in this investigation, right? The dependent variable is the thing that changes, or usually the thing that you're testing for, right? So that would be the heart rate. The heart with 2 A's. Sorry, the heart rate, HEART-RATE, right? Or slash the pulse rate, right? That's the thing that is being manipulated, right? The pulse rates, the experiment is actively making them walk for 15 minutes, which in turn is manipulating the beats per minute. Also, they're measuring, or they're trying to record or find a change in the heartbeats per minute, and the dependent variable is usually the thing that you're either looking for or the thing that you're testing for, right? So one error in the experimental procedure is the fact that there's no control, right? I never noticed any control group within this excerpt, within this blurb, right? Any good experiment has a control, because what are you comparing the pulse rates to, right? There's no before group. There's no control of people who didn't walk for 15 minutes, right? So you're just having these random pulse rates and nothing to compare them to, right? You tested for them, right? They were dependent variable, but now what are you going to do with that? Nothing, because you don't have a control to see if walking for 15 minutes really changed something, right? So there's no control group, right? Number 80 asks us to draw a line in the graph that shows the relationship between exercise and oxygen consumption, right? So A good way to think about this is cellular respiration. So what happens in cellular respiration? You're taking oxygen and you're taking glucose or sugars, and you're breaking them down, and you're releasing ATP, you're releasing CO2, and you're releasing H2O, right? So in order to exercise is the movement of muscles. Whenever your muscles move, that requires a bunch of ATP, right? So whenever you're going to have exercise, you're going to need a lot of ATP. In order to create ATP, you need a lot of oxygen, right? So as you exercise more, as your body requires more ATP, it's also going to consume more oxygen to create that ATP, right? So you should probably have some sort of straight line going up, right? And this obviously makes sense, right? Whenever you run, you begin to pant, or you breathe really heavy. That's your body taking in more oxygen so it can produce more ATP. My support for this is everything that I've just been saying. During exercise, more energy is required, thus more oxygen is required, right? You could also say that you need more ATP, you need more oxygen to create ATP, so on and so forth, right? So base your answers to questions 81 and 82 on the information below in your knowledge of biology. Student prepared 2 potato cubes by cutting a 2 centimeter by 2 centimeter sections from the same potato. Next, she determined the mass of each cube, and recorded the information in her lab notebook. She then placed one cube in a breaker of distilled water and the other in a breaker of equal volume of concentrated salt solution. After 20 minutes, she removed both of them from the beakers and determined the mass of each cube. Which statement correctly describes the effect on the mass of one of the cubes after the 20-minute period, right? So this is a problem regarding osmosis, active transport, or diffusion, right? Something like that. We have one potato with a so she's preparing potato cubes, right? And then she's placing one potato cube in a beaker of distilled water, so it's just regular water, and another beaker in a concentrated salt solution, right? So here there's more salt on the outside, right? So eventually what's going to happen is naturally when it comes to distilled water, the potato has more salt, right, than the distilled water. So what's going to happen is in order to balance out the the difference between the salt and the water, this distilled water potato is going to intake a lot of water to make it less salty, so that the salinity or the salt level in the potato is equal to that of the water, right? And the best way to do that is to dilute that salt, to add more water. So the water is going to flow into this potato. When there's more salt outside of the potato than there is inside the potato, the opposite's going to happen. The potato is going to try expelling its water out of itself in order to equalize and dilute the salt outside, so that it matches the salt content of its own that's found inside its own body, right? So what really models this is choice number 2, where it says that in distilled water, the mass of the potato cube increased due to water moving into the cells from high concentration to low concentration. Boom. We literally just described that. All other option choices are false. Number 82 says that a student placed a thin slice of potato in a drop of water on a glass slide. She added a cover slip and a drop indicator. Using a compound light microscope, she examined the slide of a potato and made the drawing below. Right? So here we have black stained structures. These are potato cells, right? The blue black stained structures labeled in her drawing are most likely what? Right, so we have a bunch of them, and we have the actual cells, right? and it's a thin slice of potatoes in a glass slide, and she's using a compound light microscope, right? So she's not using an electron microscope. She's using a pretty not that strong of a microscope. So obviously it's going to be starch grains, right? Chloroplasts. Well, that's not found in the actual potato, right? Because the potato isn't green. The potato cell, that could be found in the leaves of the potato. She was analyzing a potato leaf, but she's not doing that, right? So it can't be chloroplasts because they're simply not found there. Sugar molecules, remember that a potato is starches, not sugars, and ribosomes. You need a much stronger microscope in order to actually see ribosomes. Compared to this, the ribosome would be pretty tiny, right? And also, if these were all ribosomes, then you wouldn't be able to see the actual cell wall, right? If these were all ribosomes, it simply wouldn't make sense why you could see multiple cells, right? And it wouldn't make sense why you wouldn't be able to see other organelles like the mitochondria and the nucleus, right? So moving on to the last set of questions, 83 to 85, right? Tests were performed to help identify the person who committed a crime. Lane D contained evident DNA from evidence found at the crime scene. Lanes A, B, and C contained DNA from each of 3 suspects. Right. So what this is gel electrophoresis, right? Right. This is also called DNA fingerprinting, but I know it better as gel electrophoresis. What happens is that these are the wells, right? You put DNA samples in you cut up DNA samples, you put it in here, and then the charge from positive to negative is applied, and then slowly the positively charged DNA bases and segments of DNA move toward the negatively charged one, and what you have is kind of this array of DNA, right? Right. So number 84 says, based on the data, which lane most likely contained DNA from the suspect who committed the crime, right? So lane D contained evidence from the crime scene, right? So this is the crime scene strain, right? And the way that you can use gel electrophoresis to determine a relation, right, is if the segments match up, right? So if you're looking B and D, right, here we have this one and this one match up, this match up, this match up, this is an identical copy, right? B is the same thing as D, which means that suspect D, or lane B, contains lane B is your answer because it just contains support your answer. Well, I'm starting to support my answer here. It's because it has the same bands as lane D, right? Each one of these is a different strand of DNA, right? Different strand of DNA, and it's cut up in different ways, right? DNA, if you cut up the same strand of DNA, it's going to be cut in the same way. If you cut up a different strand of DNA, it's going to be cut up in different ways, right? But because these two are completely identical, that means it has to be the same strand of DNA, because no two similar DNA, no two same DNA strands would have these same segments and would have the same position unless it was identical, right? So that's all. Number 85 asks us to identify the lane that contained the band with the shortest fragments of DNA, right? So if we're looking for the shortest fragments of DNA, we're obviously going to look at the bands the farthest down the gel than the others, right? Why? Because look, the DNA, as I just said, DNA, right, moves down. It moves down from the wells all the way down to the bottom, right? And it's moved because it's attracted to the negatively charged bottom, right? Obviously, the heavier something is, right, the longer something is, the heavier it is, right? Why? Because the longer something is, the bigger it is, and the bigger something is, it's usually heavier, right? So long DNA strands are often going to be heavy, and because they're heavy, they're going to move slower through this gel, right? But because A has this little segment all the way over here, right, which is ahead of anything else, that means that segment A, that well A, or lane A, had the shortest segment of DNA because it was able to move the quickest, right? Because the shorter it is, the lighter it is, the quicker it's able to move, right? So lane A would have a band with the shortest fragment of DNA because it's closest to the bottom. So that's all of it. That's questions. That's both part C and D, all reviewed in a singular video. If you liked what you saw, if you learned something, if you found this helpful, please leave a comment and leave a like and consider subscribing. That being said, that's the end of this review. I hope you all have a wonderful day and good luck with your test tomorrow. Goodbye.